Now we continue with this lecture, B2 of Sensory Physiology on gustation, or the sense of taste. Well, we know that occurs on the tongue, but also in the nearby pharynx that has a mucosal lining and scattered taste buds there. The anatomy courses is where you would go into the detail of the different type of taste buds, but certainly you can see these big circumvallate ones here, and then there's filiform and fungiform, a few different kinds. But they also, I was surprised that the authors did not show the anatomical distribution of the different tastes, but we'll survive without it. You certainly can find that information easily on your own. So there are the 5 basic tastes, or sensations, excuse me: sweet, sour, salty, bitter, and umami, which is a taste associated with the amino acid glutamate, and that's been added to the list of the classical taste sensations that you might have studied in the past. And it is, of course, directly linked to the utilization of monosodium glutamate to enhance the flavors of foods. So here we see a histological section with what's called a trichrome stain. Maybe it isn't the trichrome, I don't see the green, but definitely we have an intense red and the blue. And we can see the overlying epithelial cells of the tongue. It's a stratified squamous keratinizing epithelium. You know, the surface cells are dead. That's why we brush our tongue, to get rid of those dead cells and all the bacteria and stuff that are on them. And at the surface of the taste bud is a taste pore, and we'll look at an illustration that helps us to visualize how the apical portions of all of these cells within a taste bud are associated with each other there, and the general makeup is a little bit easier to see with an illustration. But what we want to do here is appreciate that there's numerous cells in here, and it can be challenging to identify them histologically. You'd have to cut a lot of sections, serial sections, through an entire taste bud, any type of taste bud, to get to reconstruct the full collection of cells that are in there. It's a dynamic process, for cellular replacement and things like that. So it's a challenge to study these highly specialized receptive cells and how they're associated with the nerve endings. But you should be able to picture in the underlying connective tissue the nerve endings coming in here. We can't see them directly with this illustration. An electron micrograph, we could probably distinguish them. But anyway, this gives us a basic idea of what we're considering as we transition to very nice illustrative representations of these complex structures. So a light micrograph of a taste bud. Each taste bud is composed of taste cells and support cells joined near the apical surface with tight junctions. I know this is a physiology text, but I am continually disturbed a little bit that scale bars are not used to give us a feel for the dimension. But you can picture this as your tongue. You can picture those are the little things that stick up on the surface of your tongue. And on those there'd be a bunch of these, right? There isn't a single taste bud on each of the papillae on the tongue. There are multiple ones. So hopefully that gives you a feel for the dimension. So here we see that illustration of a taste bud. So as we said, there are tight junctions at the apex of these epithelial cells. They are specialized, highly specialized epithelial cells. Excuse me. So that means typically there's a turnover rate. Most of our epithelial cells do have a turnover rate. We do not have any epithelial cell that I can think of that lives our whole life with us like our neurons do. In other words, post-mitotic. Most have some turnover rate. But you're seeing the sustentacular or supportive cells. They are around the periphery, containing the whole structure, except it does not close it off at the pore, nor at the base where the neurovascular bundle exits and enters, whichever way you want to think about it. So you can see that in each taste bud you have each specialized taste sensation represented by separate cell populations, except salty and sour overlap on the same presynaptic cell. It uniquely has that terminology because it's the only cell type that actually couples the membrane potential to the exocytotic release of a transmitter. Can't really call it a transmitter here because it's not neuron to neuron. But anyway, in this sense, it's presynaptic, and the primary gustatory neuron peripheral process terminals are the targets of that. That would be the postsynaptic cell. And so we see that the other cell types, even though there might be some commonalities in the coupling, these are all G protein coupled types of receptors for these, as they were with the olfactory system. But even though they may have that in common, they differ a bit in how they send their signal to the primary gustatory neurons. And I still get the feeling that something is missing from this story because we are so able to distinguish between these taste sensations. It's kind of hard to believe that ATP from all the remaining three types, the bitter, the umami, and the sweet, targeting essentially the same cells, even though we have different pathways centrally to perceive those taste sensations and to turn them into the more complex perceptions of food taste quality, of course combined with olfaction, excuse me, that we are well aware of. However, this is the state of our knowledge of this, and it actually is different from what you may have seen in previous physiology textbooks. So sometimes with a new edition, you do get new information. So it's not always just to keep textbooks out there that cost the highest amount because they're brand new. Anyway, we see that we are going to have, in this case, the more complex type of sensory endings, as we had studied with the first presentation of the sensory system. There is an epithelial receptor cell interfacing with the environment, in this case the external environment, and it is responsible for transducing the receptive stimulus information into a signal that makes sense to the first order sensory neurons, and then their coded information, both with the action potential information, the frequency modulation and things we've talked about there many, many times, as well as their central representation in a particular pathway, is what will provide the central representation of each unique taste sensation. So the taste ligands in this case create calcium signals that release the serotonin as well as the ATP, but they're not showing an exocytosis of the ATP here, and we're safe just to leave some things out of the picture. So we see this here. We're going to go through it in a series of slides on the summarization of taste transduction. And we start on the left here, and we see the ligands activate the taste cell. So, for example, sweet umami or a bitter ligand will bind to the G protein coupled receptor, the GPCR. There is a special G protein called gustducin, obviously transducing the gustatory sensation into a cellular signal. So there's a signal transduction through some type of second messenger system, whatever it may be. It looks like it is probably going to be an IP3 and some other target, maybe through the DAG. I don't know exactly what it is, but because of the way we're seeing this diverging to a membrane event and an ER event to release calcium, we could predict what those might be. Again, because these are special cells, it might be some unique mechanism. But anyway, with the intracellular calcium concentration increase, there's some signaling that is yet unknown that brings about the release of the ATP. And of course, I forgot to go through my sequence here. What I'm supposed to do is go across and just do the ligands activating the cell taste. The taste cell. But anyway, that gives us a feel for what's going on. We'll differentiate it as we go down through 1 through 4 there. So salt entering the cell for that type of a response, the presynaptic cell as it's called, will increase intracellular calcium, and that's going to depolarize the cell. And then we have the hydrogen ion entering the cell and increasing the pH or binding to a membrane protein, or just by changing the pH nearby might alter a membrane protein. Not really sure, but anyway, there are various intracellular pathways that are activated. We conjectured on one here, and it's still not quite known exactly how the hydrogen ion would bring some things about. But what is known, because you can very easily fill cells with molecules that fluoresce when calcium binds to it, the free calcium binds to it, and then there's tools, pharmacological tools, that will block extracellular sources and intracellular sources of calcium. So this information can be dissected from these cells. They can be suspended, the animal sources most likely, suspended in vitro solutions, and these types of analyses can be performed. In the textbook, they also mention knockout mice, so they inactivate certain genes, and they're able to see how that impacts these events. So obviously the calcium is going to trigger the exocytosis, just as at any presynaptic terminal, to use that terminology, and then it is going to be associated with the ATP formation in some way, and the release of the ATP. They're showing a channel type of a release here, and we get the primary gustatory neurons responding to the input. So remember, the purinergic type of receptors, and ATP was one of the chemical messengers listed under that type. So the primary sensory neurons fire, and the action potentials are sent to the brain. So this is the same as we had shown in the prior illustrations of the graded potentials that are produced by these synaptic events here, or this type of signaling. It's not synaptic, but it still is going to produce a graded potential, and you get the summations and determination of the firing rate of the primary sensory neuron. So, in summary then, we see these common mechanisms for the sweet, umami, and bitter ligand through the ATP, but still common across the board for calcium. So it really depends upon, I always say it this way, how the cell decides to utilize a common signal as to what the physiological response of that cell, or biological response of that cell, might be. Of course, it's some way of differential gene expression that leads to unique sets of proteins that will couple the calcium to exocytosis in those cell types, versus opening channels to let ATP out, if indeed that is the mechanism. So humans and animals may actually develop specific hunger, such as salt appetite. So most hunters or people that are aware of hunters know that you can put out salt licks. If you're raised anywhere near cattle or dairy cows, you know that salt licks are commonly put out. If you've seen the shows about elephants that will go long distances to sites where they use their tusks to dig into the clay there and consume dirt, basically, are doing it because it's a site that's rich in certain minerals, and they visit it once or twice a year and stuff like that. So it's not really known how a thirst for calcium might, or a taste, hunger, for calcium might be established, but certainly it's well known that the salt sodium requirement will be a strong motivator for the behavior of a person, an organism, to seek out salt. So this ends the presentation on that sensory system, the sense of taste.